Tonight we have a three-part reflection on schizophrenia. Now, unfortunately, tonight's speaker was going to be Associate Professor Suresh Sundar. Suresh is a clinical psychiatrist with a deep interest in childhood trauma and the consequences of that for adult mental health. Suresh is also particularly interested in asylum seekers and the children in detention. And Suresh has actually been delayed up at somewhere north of here on a mission at a detention centre. So unfortunately we don't have Suresh here tonight, but you've been upgraded to a full professor. And we have Professor Christos Pantelis from the University of Melbourne, and Chris is also an honorary fellow here at the Florey Institute of Neuroscience and Mental Health. Chris is the head of adult mental health rehabilitation out at Sunshine Hospital and leads a team of over 50 staff, students and researchers, all investigating schizophrenia. So without any further ado, Chris, how would you like to relax?
Yeah, lots of images. So
lots of images, yeah. Okay.
So
I'll leave the screen nice and bright.
Okay.
And I hope you enjoyed tonight's talk.
Great. Well, thank you very much, and I'm sorry that Suresh couldn't make it. So, but I will try and cover the areas that I've been particularly interested in and involved with for many years now. And it follows on from a lecture I gave last week, the Danks Oration, in which I focused more particularly on genetics of both schizophrenia and autism. But tonight I'll focus exclusively on schizophrenia and introduce some new material as well related to ways to perhaps treat the disorder, or at least neuroprotect the brain. So I'm a neuropsychiatrist. I am a clinician. I work with patients with severe forms of schizophrenia. I head an inpatient unit, as you just heard, at Sunshine Hospital for those that have failed to respond to most forms of treatment to date. And we often take our time in terms of really trying to get those often young people better. And some of them stay with us for 6 months up to 2+ years, and some have been with us for 8+ years. So it can be a very serious condition, a very severe disorder, and for a small proportion of patients, perhaps 5 to perhaps 10%, up to 20%, the response to treatment is not as good as we'd hope. So today I'm going to give you a bit of an overview of the work we've been doing on looking at the brain in schizophrenia, and take you through how the story evolved, why we're doing what we're doing, and what we're hoping to achieve. We've been fortunate to have support from a number of grant bodies, including NHMRC, and as well as that, a number of philanthropic trusts who have been very helpful over the years in getting some of the work going. So I'm going to take you back a little bit. I started psychiatry back in 1983, and at that time I was working in London. So I'm going to show you some of the images there because they help to inform some of the questions that I've been trying to address since that time, really. And then talk about brain changes, place them within a brain maturational context, because particularly the adolescent and early adult brain is continuing to grow, continuing to mature, and show you how the brain is changing in a very dynamic way, and then perhaps try and elucidate some of the factors that might be relevant to the changes that we're seeing. I'm going to talk a little bit about neuroinflammation because it's become a very topical area that we're focusing on now, and a little bit on neuroprotection. Is there a way that we can protect the brain from some of the consequences of this illness, and then some novel ways forward? So I like showing this slide because it speaks to the importance of not only going from the bench to the bedside, but going in the opposite direction. And the work that I've been doing has been informed by observations at the bedside And I'll show you this video in more detail later on. And many of the observations here have led to questions that we've addressed with brain imaging and more recently with neuropathology, looking at the cells in the brain, genetic work which we're embarking on increasingly, and then attempts to identify new targets. And we're working here with the Florey on animal studies informed by the observations that we've made at the bedside. So a lot of the work I'll be telling you about relates to brain imaging. These are the kinds of brain images we can generate from magnetic resonance imaging. Magnetic resonance imaging that provides very detailed structure of the brain. We can look at the structure, the grey matter, the white matter here, tracts, the morphology or the folding pattern of the brain. We can look at the in vivo neurochemistry of the brain, looking at the metabolic peaks. This is a sort of spectrum you get when you go to the airport and you're assessed for drugs. They get metabolic peaks that point to particular types of compounds. They're obviously looking for drugs. But in the brain, we can do the same thing and look at different metabolites because they're informative. We also use functional imaging where we look at blood flow in the brain. And we look at blood flow either when people are just spending some time resting, just thinking, or when they're engaged actively in tasks. And that kind of functional imaging allows us to work out which brain regions are working whilst people are undertaking various high mental tasks. Nearby, just in a few doors down in this building, we have a new 7 Tesla MRI machine. Now what does that mean? That's 7 Tesla is a very high field strength MRI magnet. The ones we've been using down here have tended to be either 1.5 or 3 Tesla machines. Still much stronger than the magnetic field of the Earth, but the 7T is particularly strong and allows us to get at really quite detailed, make really quite detailed observations of the brain right down. It's almost like looking at pathological specimens. So we're just working on that now. But just to give you an idea of the complexity of the problem, both from an anatomical perspective and a genetic perspective, the brain consists of 100 billion neurons. We have 100 billion neurons at birth, so therefore important to neuroprotect and protect those cells, if you like. At birth, they're not necessarily connected, and over the next few years after birth, you see a massive proliferation of the connectivity, as you can see here, in which those neurons wire up. They also myelinate so that the speed of transmission increases. And you end up with 100,000 miles of axons and 100 trillion synapses. They're the little connections between different neurons and axons, etc. And they're very dynamic. They're changing minute by minute and second by second. So as you engage with people, with activities, etc., those synapses are also changing. And those statistics there give you some perspective on what we're dealing with. So we're trying to get a handle on what's going on in this brain. The brain is also metabolically a very active organ. At birth, 87% of the body's metabolism is allocated to the brain. That's a huge amount if you think about that. It gradually diminishes over time as the brain myelinates and as it wires up and it becomes more efficient in the way that it does things. There's a pruning of the synaptic tree that you have there. But it's still quite metabolically demanding even in adulthood. 25% of your metabolism devoted to the brain. So when your kids tell you that they're sitting there and they're working really hard, Dad, and they say, well, I don't need to go out for a run. I'm going to show you later that's not quite true, but they certainly are using energy. Now, when we're trying now to link genetics to that complex picture related to the brain, and the genetics itself is equally, if not more, complex. And quite extraordinary when you think that these bases here and these pairs of bases, guanine, cytosine, adenine and thymine, form the basis of our DNA, of the double helix that you see here, identified by Watson and Crick. And then when you scale it up, you see that there are about 3 billion of these base pairs in our genome. I like the statistic there, 2 billion copies of War and Peace. Chad Bousman gave me that statistic. And as well as that, it's polymorphic. That means that the base pairs vary. They can vary at particular locations within the DNA. And they vary by ethnicity, they vary by various diseases as well, and they can represent markers related to particular diseases, and we use that polymorphism in order to identify potential markers of illness, and I'll show you that in a little while. When you then start to look at the way that these genes are expressed in regions such as the brain, it starts to get very complex. And there are a number of different pathways. Each one of these is a pathway in the brain, and there are many more. And a number of genes code for those pathways in the brain. It provides a way possibly to look at how things go wrong in the brain. And you see an overlap between various disorders: autism, attention deficit disorder, schizophrenia, or intellectual disability. And looking at the overlap in the expression of the genes presents us with a very complex map. We're trying now to link the brain findings to those genetic findings. So let's go back to the clinic. So here's where I started psychiatry. This is Colney Hatch Asylum, or Free and Barnet Hospital. It was in the 1800s when it was built. On the outskirts, it was outside London, in fact. It's now part of North London. It's been turned into yuppie apartments. Quite institutionalised, actually, even now. But you see, it boasted the longest corridors in Europe, miles and miles of those, and it gets quite cold in the winter, which was unusual for a young Australian going over, seeing that and seeing patients that end up in a stuporose posture because of their illness sometimes wander out into this cold, and one patient had lost their toes. So this is what I'm going to show you now is try and give you a little bit of an overview of schizophrenia and then the way that we're trying to understand it. But it was observations in that asylum that started me thinking about what's going on in the brains of these patients with schizophrenia, and many of them had been there for 20 plus years.
It's very difficult to be sane one day and the next, to be hearing voices for the very first time.
Oops. Now that was Tom. He was one of our patients. He's on one of the drugs that's most effective in patients with resistant schizophrenia, the patients that I treat. He did reasonably well on that drug and was able to go out into a community care unit. But he describes there the experience of hearing voices for the first time. You can imagine if you're hearing voices in your head but experience them as though they're real and that they're coming from outside. You believe that these voices are coming from somewhere else, not from inside your head. You hear them through your ears. So it's a bit like, and I was trying to get an idea of what this might be like. The kinds of voices that our patients hear are voices in the third person. And what that means is that they hear a number of voices that are talking about them. You can imagine how disconcerting that might be. Even when we hear people talking about us in real life, it's disconcerting, let alone hearing voices that don't belong to anyone, talking about you. They may be commenting on people's actions, and they can also hear command hallucinations. Command hallucinations can be very worrying because sometimes people act on those, the voices telling someone to do something. So I tried to think about how we might emulate this. So this is sort of multi-speaker babble. It was at an art exhibition in California that I visited. And just think about trying to hear, trying to attend to talking to me or listening to me whilst this is going on in the background, and the person is hearing that. And these voices can be very distracting. So a patient I saw 2 days ago is completely preoccupied by voices in his head, and he has great trouble he had great trouble attending to the interview, and he'd periodically look up and appeared to be hallucinating. The other characteristic feature of schizophrenia is abnormality in thinking or disorder in thinking. And you can see here, this is a transcript from another one of our patients given to one of our researchers. And here, when you read some of the things he's written here, this is premorbidly a highly intelligent person. It's internally inconsistent, it's illogical, it's also grandiose. He has grandiose delusional ideas that is important. He has various degrees. He's a fellow of various medical colleges. He's the forefather of most of the Australian population, it would seem, including the Aboriginal population. All entirely illogical, inconsistent. And when you see patients with this kind of abnormal thinking, one, it's very characteristic, and two, it's difficult to understand how they can, how they piece this together in their own mind. And when you're listening to patients, it's very difficult to remember what they've said when they experience this kind of thought disorder, because it's not logical. So the disorder affects young people in the prime of their lives, late adolescence, early adulthood. Women get it a little bit later, and it's thought that there may be some protective effects of estrogen that helps. And you can see there that the incidence, or the number of new cases per year, means that in Australia about 2,400 people will develop a schizophrenia-like illness for the first time in Australia. And if you contrast that with childhood schizophrenia, which is a very rare condition, we have probably perhaps 50 cases in 1 year. So difficult to study childhood schizophrenia, though we've collaborated with some researchers in the US, and I'll show you some of that data. But what this means is that the disorder is a disorder of young people in the prime of their lives, in late adolescence, early adulthood. And that, I think, is important to reflect on, because at that time, the brain is changing in very particular ways. It's also a very heritable disorder, so the heritability, which relates to the genetics of the disorder, is said to be around 80%, so highly heritable. But it's not one gene, it's not one genetic or part of a gene that is relevant to developing schizophrenia. Rather, it seems to be a combination of genes. It's polygenic. So there are a number of genes that come together to cause this disorder. And certainly the more recent genetic work is looking at polygenic risk. That doesn't mean that all those genes identified are relevant for any particular individual, and it may be that within families or particular groups, a smaller number of genes are relevant. You also get interaction between genes, which makes it even more complicated to try and unravel what's going on. And the interaction of gene and environment is also important to take account of. When we look at the disorders, mental health disorders, most of them commence in adolescence and early adulthood. So you can see here psychotic disorders and schizophrenia, late adolescence, early adulthood, depression, similarly, substance use disorders, and then some childhood disorders. which occur at an earlier stage of development. So what is happening here in adolescence, what is happening during this period is important to think about when trying to understand the brain changes or the emergence of the disorder. Here's another way of depicting the same kind of thing. This is the burden of disease related to when the disease begins. So mental disorders have their greatest burden, lifetime burden, at this early developmental stage. Neurodegenerative disorders, of course, like Alzheimer's dementia and other forms of neurodegeneration occur later, and they have their greatest burden up here. So you can imagine if you develop schizophrenia, if you develop a chronic form of the disorder, it can be very disabling for a whole lifetime. So efforts to try and ameliorate, prevent, or at least reduce the impact of these disorders is really quite important. There are a number of brain regions that are considered to be important: the frontal lobes of the brain and the temporal lobes of the brain in particular. The frontal lobes are interesting because they form 1/3 of our brains. The human brain consists, 1/3 of the human brain consists of the frontal lobe, so the front areas above the eyes. It's an important area related to higher level mental function, cognitive flexibility, higher level executive functions, planning ability, the ability to inhibit inappropriate responses, which is thought to be related to this region just above the eyes. High level emotional processing, also relevant to this orbitofrontal cortical region. The cingulate connects many of these frontal regions, and it seems to be relevant to attention and directing attentional processes. And I'm going to talk a bit today about the hippocampus. This is a region deep in the brain. It's linked to what's called the limbic system of the brain, which is to do with emotion and behavior, and the hippocampus is important to memory, and in fact important to making associations. So when you make associations between items or things that you need to remember that are not necessarily related, it's the hippocampus that's particularly active. And this region is affected in a number of conditions. So the frontal lobes, as I mentioned, 1/3 of the human brain, which is very different compared to other lower species. So the frontal lobe is the most recently evolved. It's also the last to mature, and it tends to mature in adolescence and early adulthood. So the processes going on at this critical time when these disorders are occurring implicate this region, and probably because it's maturing late. And when we look at brain scan studies, put them all together in this meta-analysis, the frontal lobes come up quite prominently, temporal lobes come up, and there's also some regions in the subcortical area close to the hippocampus, but also the insula, which is important for sensory information coming into the brain. So what happens at earlier stages of the illness, and is there evidence that these changes are progressing in some way? And this has been quite a topical area because for a long time it was thought that things were pretty static. But the work that we've been doing has really focused attention on the importance of mapping trajectories, looking at things over time, not just at one point in time. So for instance, if you were to look at these three individuals at this point, let's say at this particular age, and you measured some sort of brain index or physiological index or something else, they may look very similar, and you might incorrectly conclude that they're on a similar path or have a similar problem. And it's only when you map their trajectories over time that you see that they've got very different trajectories. And indeed, in schizophrenia, we now have data for each of these trajectories. But let's go back a little bit historically. Whilst people for a long time thought that progressive changes didn't happen. It's perhaps that they didn't look all that well back at the available literature. This is a study in which was a series of studies done from 1935, 54, and then again in 63. What the investigators did here in patients with schizophrenia was to put a needle into the spinal cord area where the cerebrospinal fluid was. They'd extract a little bit of that fluid. They'd then replace that with some air. And then they'd place the patient, get them to lay down. The air would go up into the ventricular system of the brain, and then they did an X-ray and looked at these spaces in the brain, which are normally filled with fluid. And here you can see the ventricles here are enlarged. This is a very painful procedure, and it leads to headaches which can last weeks or even months. In these studies, they were undertaken twice in these patients, so certainly not something you'd do now. But what they demonstrated was that over time, there was an increase in the size of the ventricles, suggesting progressive change, potentially progressive deterioration. It wasn't until brain scanning, computer tomography, and then MRI came in that we started to get the kinds of pictures that would allow us to really look at these kinds of things in more detail. And here is one of the first studies published in The Lancet by these prominent researchers, Tim Crow and Eve Johnson, both who've had a great impact on the field, showing that the ventricles were enlarged in patients with schizophrenia. But there were a lot of problems with this study, and it certainly wouldn't be published now, but it certainly highlighted attention and got everybody thinking about what's going on. And this is another important figure in schizophrenia research, Danny Weinberger, and I was fortunate to work in his lab in Washington during 1992 before returning to Australia, and that is indeed where I started my imaging work. And he undertook some important studies published in the New England Journal of Medicine showing that the ventricles were enlarged in twins where one twin had schizophrenia and the other didn't. It was the twin that had the disorder who showed that ventricular enlargement. This is Danny more recently. He's been given a huge $100+ million grant to set up the Lieber Institute at the Johns Hopkins University. So I visited, took a number of pictures of Danny. He was certainly very excited by all the new toys that he's been able to acquire and get going. He's been doing some fabulous work. So here are the data from that study. And in fact, the paper itself said that you could visually identify who had schizophrenia by the size of the ventricles. Now, that doesn't happen very often. You see it in twins potentially, but when we look at patients with schizophrenia, the ventricular size doesn't discriminate adequately between normals and those without. Certainly, it's enlarged at the population level, but at the individual level, it's more difficult. So it's the issue of progression over time. And so Danny proposed in this seminal paper in 1987 that there was a fixed lesion from early in life. This paper drove the field for the next 20 to 30 years, in effect, 20 years, really, in which people tried to identify when during fetal development the brain started to be abnormal. They were focused particularly on the second trimester during fetal development, because we know that during fetal development things like influenza, things like hypoxia, other kinds of difficulties during gestation can adversely influence the fetus and increase, at least slightly, by 1 to 2%, the risk for the later development of schizophrenia. But when you look at the data more closely, it's not quite as easy as that. And it seems that any kind of insult at any time during development, before birth, during childhood, during adolescence can increase your risk for developing schizophrenia. But it was that paradigm that really started us thinking about what we might do with imaging. Now, Danny's work focused particularly on patients with established schizophrenia, but we were fortunate in Melbourne to be working with Pat McGorry and Alison Jung at the Origin Research Centre in the early days when they were developing their high risk for psychosis strategy. What they did was to identify young people who were presenting with problems, difficulties at school, their behaviour had changed, their level of functioning had changed, they were no longer doing as well as they should have been. And they found that there were a number of factors that characterised these young people. Some of them had attenuated psychotic symptoms, like hearing a voice for a day, but certainly less than a week. They may have had family history of schizophrenia, but certainly they had a drop in function. And almost 40% of these young people became psychotic over a 12 to 18-month period. What this meant was that we were able to assess these young people before any of them developed illness. We did a brain scan, we did cognitive testing, we did clinical assessments, and then we followed them over the 12 to 18-month period and looked to see who became unwell, and we scanned them again. And in the first study that we did, we were motivated by Danny's work, Danny Weinberger, in which we looked at the hippocampus. So in Danny's paper, they looked at the hippocampus in the twins, and it was smaller in the affected twin. So the twin with schizophrenia had a smaller hippocampus. As I mentioned earlier, the hippocampus is important to memory, particularly making associations, important to behaviour, and it's part of the emotional brain, the limbic system of the brain. So our first work was really to look at the size of the hippocampus in patients with schizophrenia, not only in those with established disorder, but to go right back to before the illness began. Our hypothesis was that we could use this as a marker to predict who would develop psychosis. So if Danny Weinberger's hypothesis was right, that it is an early lesion, then we should identify it before anyone became unwell. So here are the data. Here are the data in chronic schizophrenia patients. They had about an 8% reduction on both sides, left and right hippocampus. But then when you went back to earlier stages of the illness, in first episode schizophrenia, the left is down but not the right. And I haven't got an arrow for this one, but these are first episode psychosis patients within the first 6 months of illness. And that's trending down but not significant. But more importantly, when we go to the pre-psychotic high-risk group, so in other words, we did a scan before anyone developed psychosis, followed them over 12 to 18 months, and this is the group that became unwell. Their hippocampi were not smaller. We also did memory testing, and I'm not showing that because of the interest of time, but it paralleled these findings. So it looked as though brain structure in the hippocampus, as well as brain function, was not abnormal at the very beginning of the illness. Now this is really quite important because one of the notions about schizophrenia was that it was you're doomed from the womb, and that was based on that original hypothesis. Clearly, if that's not the case, we can make a difference at this early stage. Perhaps we can prevent these brain changes occurring if they're progressive. At that time when we did this study, we didn't know if the changes were progressive. But these data specifically led us to do studies longitudinally in a number of our groups of patients, but most particularly the high risk for psychosis patients, those in which 40% developed illness over a 12 to 18-month period. And here is the first study published in The Lancet, in which we were the first group to demonstrate changes in the brain dynamically as young people develop psychosis for the first time. So the frontal lobe and the temporal lobe were showing significant changes over time. The cingulate was also involved, relevant to attention. So this was the first demonstration, and this paper has generated a great deal of interest. It's now a highly cited paper and has generated work across the globe looking at this high-risk group from an imaging perspective and trying to identify what's happening. So that work, those two initial studies, really set us on the path to looking at trajectories, to thinking about trajectories in the brain, to thinking about how other brain regions may be changing. So now here what you see is some work by some colleagues that we've done, some work that we've worked with over recent years. Nitin Gogte, Judith Rappaport and the team in Washington have undertaken this fabulous study, started around the time when I was in Washington actually, and now they've got data since 1991 or so, in which they scanned young people from age 4 to age 21. These are the data showing, I don't know if that's moving, it's meant to be a movie. Yeah. So what you see is a gradual loss of the brain's, a thinning of the brain's grey matter, starting at the back of the brain, moving forward and downward, finishing in the frontal lobes and also in the temporal lobe, finishing in late adolescence, early adulthood. So this is a normal maturational process. The normal adolescent brain is thinning slightly, and it's thought that's to do with a pruning of the synapses to make the brain more efficient. So losing grey matter is not necessarily a bad thing. It makes the brain more efficient, it seems. Sometimes. But when we look at schizophrenia, what's interesting, and these are data in which we scanned people just after they became unwell for the first time, within the first few weeks and months, and then we followed them over 2 to 4 years. And we looked to see where the grey matter went between, did it move towards the skull, expanding, or did it move away from the skull, implying a retraction or thinning? And there was retraction in this frontal region in first episode schizophrenia. These are the normal, healthy individuals showing this normal pattern. And when we compared these two and mapped one against the other, the rate of loss here is going twice as fast as it normally should. So what we're seeing is a normal brain maturational process that's accelerated twofold, going twice as fast as it should. Potentially there are genetic factors that might be driving this, and we've got some clues to some of the genes that we think are important at this early stage of the illness. And then we looked at this, our high-risk group again, and we see the same pattern of frontal lobe thinning, which occurs as people develop psychosis for the first time. So this process has already begun prior to onset of an active illness and seems to continue for the first 2 to 4 years following onset of the illness. If so, then we should be really targeting to try and prevent this happening, and it may have important implications functionally. So what about the brain's white matter? So the brain, as I said, at birth has 100 billion neurons. They're not particularly wired up, but during the first year of life, there's a massive proliferation of connections, of myelination across most of the brain, except for these regions that are developing in adolescence and early adulthood, the frontal and temporal lobes, and particularly the frontal lobe.
From age 12, the grey matter, the thinking part, is being pruned, keeping only the most efficient connections. Meanwhile, whole brain sections are being coated in a white substance called myelin. This white matter speeds up nerve conductance 100-fold, creating fast brain superhighways.
So in effect, the axons that connect the neurons are being insulated by the myelin. And that means that conduction of electrical signals across the brain increases in speed very dramatically. and that makes the brain more efficient, faster, we think faster, etc. So we employed an electrical engineer in our group because we thought, well, if you want to look at the wiring of the brain, and someone with skills in looking at wiring might be useful. It was a bit of a gamble, but he's done an absolutely fabulous job in terms of looking at brain wiring. And this is the work of Andrew Zaleski. He just finished his PhD when he joined our group some years ago now. And you can see he's generated these pictures where he looks at the connectivity between different brain regions and is able to identify the streamlines connecting different neurons across the brain. And in this study looking at schizophrenia, and you've got the cover for this, we find that these frontal and temporal regions show abnormal connectivity. We were interested that in the back of the brain we see some similar things as well, and that is an evolving story at this stage, but certainly frontal and temporal areas show this abnormal wiring pattern. So we're seeing changes in the brain's grey matter, we're seeing changes in the brain's white matter, and the frontal and temporal lobes are key regions implicated in the disorder. So what about the genetics, and how does that help us? In fact, I'm just wondering whether I have, and I'll come back to the genetics, in fact. We've also been looking at the heritability of brain connectivity. So what are the genetics of brain wiring? And these are normal twins in which we found that the brain's wiring is highly heritable and related to the efficiency with which the wiring works. We haven't yet unravelled what the genes are relevant to this, but that it will be important for us to undertake in looking at a disorder like schizophrenia, where the wiring and the connectivity are impaired. This has become a very topical field, in fact, the human connectome, which received a lot of funding from NIMH to try and map the brain's connectivity. So it seems the brain is changing in structure in early psychosis, and that is associated with functional changes as well. There's evidence for neuroprogressive changes, and again I stress the temporal and frontal lobe regions. Some of these changes reflect accelerated normal developmental processes, but others suggest neuroprogressive, potentially degenerative processes that we don't know really what the basis of that is yet, and there may be a genetic contribution. But is there any evidence for improved trajectories? I mean, all that sounds like a very negative story, in fact, but we've turned our attention to looking at whether trajectories might change in a positive way. And this is some work that we did in childhood schizophrenia. As I mentioned, a rare disorder, and that we were able to do this study with our colleagues in Washington because they were gathering data on children with schizophrenia. And because of the much larger population, this was possible there. They would fly the families in from all over the country. They would house them. They would treat the children and help the families, etc. And over a period since 1991, they've been able to undertake serial scanning every time the child came back. Not only that, they were able to also do brain scans of the unaffected siblings. So the brothers and sisters of those children who didn't develop the disorder. And this is what this analysis is about. Here we're looking at brain connectivity, the connectome, and it's looking at measures of that based on grey matter measures. It's called structural covariance. What you see here in red is the children with schizophrenia, and they have an abnormal pattern of connectivity right through the adolescent period into early adulthood. They start to normalize a little bit here. The blue line are normal, typically developing children around the same age into early adulthood, so from age 12 to 24. The black line are the unaffected siblings. So what's interesting about that is that the unaffected siblings show a very similar abnormality at age 12. But by mid-adolescence, by age 16, they're normal. A better way to show this is in the following movie. So this suggests that there's an improvement in brain connectivity in the unaffected siblings. So presumably they're at risk or show some of the risk markers for the illness. They don't have symptoms, but they show the risk markers. And over that initial adolescent phase, they catch up to normal controls. So here's a movie of the same or similar type of thing. And what you see here is a childhood schizophrenia. The numbers represent the times of the scans, when the scans were done. And here is the unaffected siblings. Abnormalities at early age. And here, the unaffected siblings initially show the abnormality and then it goes away, in effect, or at least they're not different to normal. So that, we think, is important and may speak to the relevance or the importance of resilience factors developmentally. So are there factors, genetic or otherwise, that lead to resilience, one from the illness, and two, in terms of these brain changes normalizing? And if so, can we identify those, because they may be particularly important in helping us to improve the function and the lot of many of our patients. So we didn't stop there. We started to then think about our high-risk group. I don't want you to focus too much on the details in this slide. I realise it's a complex slide. But what we did here was to look at white matter connectivity. We looked at high-risk individuals, scanned before anyone became unwell, and then followed them over 12 months. And rather than focus on the group that became ill, we focused on the group that didn't. What we found was that the white matter tracks improved. They got better over the 12-month period, and their symptoms diminished, suggesting that whilst the group that became unwell had the opposite type of pattern, this group was showing an improvement, a reduction in symptoms, and as well as that, an improvement in the integrity of white matter in the brain. Again, speaking to potentially to the issue of resilience factors in the brain. So these changes suggest a number of potential processes, but it's important to try and reflect on whether we might be able to understand what might be causing these changes. Is there any way that we can get a handle on that? So I'm going to talk briefly about genes and stress, cannabis, and a number of other factors, just briefly. So here are the genes, the base pairs that I mentioned earlier. This here you see the polymorphism that I mentioned. This base pair gets replaced by this, and this varies by population. And these polymorphisms represent potential markers that we can index relevant to a particular disorder. But it's important to be clear about ethnic differences when undertaking these studies. So here is the largest genome-wide association study based on 500,000, I think it is, SNPs or polymorphisms in those individuals, over 50,000 individuals in this study, almost 40,000 patients with schizophrenia. So this is a multi-centre study that we participated in as well. We contributed data from our patients. There are 300 authors on the paper because of the number of centres that were involved. It's an important study. It identified 108 genetic loci. The challenge now is to try and understand those. But this signal here at a very high significance of a p-value of 10 to the minus 30. For many of our studies, we look for a p-value of less than 0.05. This is a p-value of 10 to the minus 30. And this signal is related to immune function. And it's really led us to start thinking about immunity in schizophrenia. This is related to dopamine D2 receptors. Dopamine D2 receptors are where our antipsychotic drugs act. So everybody was pleased to see this signal. So we can look across all of those SNPs, all of those polymorphisms to get those kinds of signals. We can also look at individual polymorphisms and how they may be relevant to the disorder. We've been focusing on one gene, neuregulin, on chromosome 8, and we've looked at polymorphisms, the ones in red we're focused on, in that gene. And what we found in the high risk for psychosis group was that those developing psychosis, each step down here is a person becoming unwell, those polymorphisms in this neuregulin gene predicted who would develop the illness, suggesting that genes are important for developing the disorder. And neuregulin is relevant to a number of neurodevelopmental processes, including brain development, myelination of white matter, and many other processes, but it also potentially relevant to inflammatory processes. So that is looking interesting. We've pursued these data further, and we've looked at the influence of this gene, neuregulin, and those SNPs in particular, on brain structure. And we see that ventricular size, which I showed you earlier, is more apparent with certain genotypes or allelic variation in the neuregulin gene. We also see relationships with white matter tracks as well. So it's looking like a promising candidate for us to look at, related to the onset of the disorder, but also in relation to brain structure. What about stress? When people are developing psychosis for the first time, it's a very stressful time, a very stressful event, not only for the individual, but also for their families. And stress is an important factor in people becoming unwell. And I think if there's one thing that we can do is for young people that are at risk, is to reduce levels of stress, I think. What we did here was to look at the size of the pituitary gland. The pituitary is relevant to the stress system of the brain. And it's related to cortisol levels, so the stress hormone. And we looked at the size of the pituitary with the hypothesis that if stress is important, the pituitary should be larger. What we found that yes, it was larger, and then when we looked at a predictive model and we divided the group into those with large and those with smaller pituitaries, it was those with larger pituitaries that were developing illness. So again, each step here is a person becoming unwell. If you have a larger pituitary, you're more likely to develop the illness. And the size of the pituitary is correlated with how close you are to making the transition to illness. So we're now pursuing stress and the importance of stress in the onset of psychosis. What about cannabis? We know that many of our patients use cannabis. In fact, probably up to 70 or 80% are cannabis users. And cannabis is implicated in the onset of psychosis and probably precipitates the onset in those who are vulnerable. It doesn't affect everyone in the same way, but if you're vulnerable to the disorder, perhaps from a genetic predisposition, you're more likely to develop a psychotic illness. So here we looked at not ill people, but rather, well, supposedly healthy individuals who were long-term heavy cannabis users, using cannabis for greater than 10 years, greater than daily use or daily use over that time. And we found that the cumulative exposure to cannabis was associated with a reduction in the size of the hippocampus and the closely associated amygdala, which is important in emotion and fear. So these structures that have been implicated in schizophrenia are being affected adversely by cannabis. Not only that, when we look at the white matter tracks across the whole brain, in a larger sample of heavy cannabis users, it's the white matter tracks close to the hippocampus that are relevant. So it seems that cannabis is having a very specific effect on the hippocampal region of the brain. And these people have difficulties with memory. Some of them have psychotic-like experiences as well, but have never been diagnosed. The other interesting thing is that certain genotypes in genes like COMT, which is important to dopamine, the target for many of our drugs, this particular genotype, if a young person has that genotype, VAL, and they start using cannabis during adolescence, it seems they're more likely to develop a psychotic illness. So here we see an interaction between the timing of onset or onset of using cannabis and a gene and a genotype in an individual might predispose them to developing the illness. So delaying use of cannabis in those having that genotype might be important. And there are other genetics that have been identified as relevant in a similar kind of way. We've been looking also at antipsychotics. I guess I'll have to finish fairly soon, so I might rush through some of this a little bit. So many of you might be familiar with some of the drugs that we use to treat schizophrenia. We have two epochs of antipsychotic medication. Those developed in the 60s, such as chlorpromazine, haloperidol, clozapine, which is deemed an atypical drug, was developed then, but only came back into use more recently. And more recently, we've had a second generation of antipsychotics that don't cause as many side effects related to movement disorder or disorder of movement. I'm going to show you a video of a gentleman who was assessed in the 70s on a very high dose of haloperidol. Normally we'd use 2 to 5 milligrams. He's on 30. And you can see the effect.
I was sleeping or something. It's being used. That this exploding.
You can see he's got Parkinsonian tremor. He has difficulty sitting because he's got akathisia or inability to sit due to the medication. He's got tardive dyskinesia. He's got sustained contracture around his muscles of his face. And he's got a dystonia affecting his tongue, making it difficult for him to speak. So in effect, this is what people often complained about, that these drugs lead to people ending up in a chemical straitjacket. And this is the sort of thing that you saw in those early days. And the dose was pushed up because it was hard to control the symptoms in some patients. And because they were relatively safe, and I say relatively, the dosages seemed to go up to too high levels. So the question is, does some of this medication lead to some of the brain changes that we're seeing? So we undertook our study in our first episode group, and again, I won't go into the details here. I think the second slide shows it a bit more readily. But we looked at the effects of medication, we looked at the effects of the illness, and we found that they were different effects. So the illness is causing some brain change, and the medications are causing some brain changes. And I think you can see them a little bit more readily here. The blue are people who are on those first generation drugs like haloperidol. In each of the graphs, this left-hand column is normal, healthy individuals. These are the ones in the blue who were on the first generation drugs like haloperidol, with showing a thinning in different brain regions. And by contrast, our newer drugs don't show this thinning. If anything, they're a little bit above control. So there seems to be a difference and perhaps a better impact, though that's still unclear with regard to the second generation drugs. The second generation drugs have their own problems, I might add. They lead to obesity, diabetes, high lipid levels, and the metabolic syndrome. So our medications are okay in treating the illness up to a point, reducing the side effect burden on our patients is a key issue in the treatment that we're constantly tackling. So medication may be important, and we need to be thinking about that and thinking about it very cautiously. Now, I know that we're running a little bit out of time, so I'll go a little bit more rapidly. We were interested to know whether the trajectories were smooth or whether they actually were episodic. There may be times when people relapse with their illness when the brain is changing more dramatically. There's very little research that's been done to date on this, but there's some evidence to suggest that as people relapse, their brains swell a little bit. And then when they get better, their brains reduce in size. That might indicate inflammatory processes, which is something that we've been investigating. And we see here that the most dramatic changes are in the earliest phases of the illness. So that's a very active time when you have more acute relapses and remissions. And we have some data here showing an increase in this signal T2 on our scan, and that index, the T2 indexes water content in the brain. So if you've got a bit of swelling, you're going to get an increase in that signal. And we see that in the hippocampus here. And I'll come back to that in a little bit. So we wanted to explore inflammation because it potentially might explain some of the brain changes that we're seeing. So we're interested to look at the brain's immune cells, the microglia. There was a little lesion induced here, and then you see this influx of these microglia. What the microglia do is in their normal resting state, they're like this, and they're purveying the environment, assessing it to work out if there's any trauma or damage or some kind of insult to the brain. If they detect an insult, they retract their processes, they become aggressive and amoeboid-like, they descend onto the region that's had that insult and try and contain the damage. Quite useful if you've had a stroke, if you've had a neurodegenerative disorder, etc. But in patients who are developing during adolescence, this may not be a good process if it's gone awry. So we've been looking now using positron emission tomography at tagging these cells, the microglia. And this is really still preliminary findings. We've now scanned almost 60 individuals. We've yet to analyse all of that data, but there's some suggestion of differences, though it's going in a counterintuitive way, which we think might be related to treatment, but we're not sure about that as yet. So mapping the brain is important, and there's a number of things that might influence the way that the brain is changing. Genes, stress and inflammation, activity levels, in fact, may be important, medications and illicit drugs. I'll finish off with ways forward and go fairly quickly on this, but one of the things we can do is take these various imaging modalities and combine them, and we've started to do that in studies like this, where we look at the connectivity of the brain, both in terms of brain structural connectivity and brain functional connectivity. And this study shows that those two processes have fallen apart in schizophrenia. The normal link between those processes is not going correctly. Neuroprotection, I just want to touch on that because it touches on what I've just been talking about with regard to relapse as well. And here again we're looking at this T2 signal which is picking up potential swelling in the brain. We see that the T2 signal is larger in the hippocampus before the onset of illness, and if you treat with a low dose of lithium, half the normal dose you'd use in bipolar disorder, you see what seems to be a neuroprotective effect. It reduces that swelling. We've yet to elucidate exactly what that process is. The other treatment that people have been trying recently is the use of eicosapentaenoic acid, which is a component of fish oils. And it's thought that fish oils are protective. So what we did was to do an in vivo study, or a study looking at in vivo chemistry of the brain, focusing again on the hippocampus and looking at this peak here, which relates to antioxidants in the brain. And we see an increase in these antioxidants with this fish oil treatment. Got to be cautious. One of my colleagues mentioned that if you're thinking about fish oils, you've got to be very careful about what you buy, because you've got to be cautious about the mercury content of fish oils. You've got to be cautious about the amount of EPA, or icosapentaenoic acid, because that's the active ingredient. And there aren't many versions or manufacturers that get it right. So buying any kind of fish oil is not necessarily the right thing. I want to touch on exercise because it affects all of us. And I've been focusing a lot on the hippocampus. It's a key area because it's very susceptible to all manner of things, environmental as well as potentially genetic. But here, this is not our study. This is a study by colleagues in Munich and elsewhere. And what they did was to look at exercise and the impact of exercise on the hippocampus. The hippocampus is important because one of the key places where you get the birth of new neurons is in the hippocampus. And exercise increases the size of your hippocampus in normal, healthy controls. So you've all got to go out after sitting here for over an hour, and do some exercise, okay? It's important. And it'll help your cognition, your memory, potentially at least. But there's some evidence showing you that the hippocampus improves in size. And in schizophrenia, you see the same effect. Hippocampus increasing in size with exercise. So an important rehabilitation strategy. We've been working with people here at the Florey, taking our data on cognition, I haven't shown you much of that, and translating it to animals, and we're doing interesting studies that I didn't think mice were capable of doing. And here you see a mouse getting a pellet of food or something, comes over to this touch-sensitive screen that touches with its nose. It has to select the correct pattern, and if it gets it right, it gets some food. They're very clever, actually. And mice and rats have large hippocampi, so if you're interested to understand the hippocampus, they're a good model. So I'm going to go over this. I'm not going to show you the video because we're running out of time. But one of the things we're doing now is looking at children with schizotypy or schizotypal features, and we're working with child psychiatrists and child neuropsychologists who have identified a group of young kids, about age 8 to 10, who have imaginary friends and imaginary worlds, and they often draw these imaginary worlds, and they often have various types of figures in them. I do have a video. Is there time, or have I? Yeah? Okay. So this is a video that's a child's film called Shark Boy and Lava Girl. Does anybody know the movie? Anybody? No? Okay. I think this shows someone that shows the kinds of features that you see in these schizotypal children. They have imaginary worlds, imaginary friends. They often get bullied at school. They're often preoccupied, so they live in their head, and they find it difficult to sort of think about other things. They keep a diary as well, often.
He had been traveling the universe in search of his father. He told me of his latest adventures on a planet so cool, it makes you drool. He said I too could go to planet Drool someday. Then one night, I was visited by a glowing light. An amazing girl with purple flames for hair and skin of molten lava rock appeared. I called her Lava Girl.
It's interesting, the film does go on to show that at school he's bullied, the teacher doesn't believe him, his fellow students don't believe him, etc. So how are we looking at these kids? Well, we've done an initial study looking at cognition, but I think what's really interesting is this that we're going to do. And this is some work that one of my researchers has done, Ben Harrison, looking at how the brain is active when you're just thinking. So you're not focusing on the external world, you're just deep in thought, just allow your thoughts to sort of go freely. And you see activity in what's called the default mode network, and these are the regions of the default mode network, including the frontal lobe, including cingulate and other areas. And what Ben was able to demonstrate was that when someone started to engage in a task, a cognitive task where they had to focus outside themselves, that network there was deactivated. By contrast, if they were in that resting state, just thinking, it was active. And it also became activated if they were focused internally. And for that he used a mood induction paradigm. So he got them to think about things in their head. And during this internal preoccupation, that network was activated. So what we're looking at with these children is to see whether this default mode network is the default state, if you like. They tend to switch back to that network. And we've got some preliminary data in our kids with schizotypal disorder. You can see it's a very similar network being activated here. And we're also looking at kids with autism. It's only showing one. We've now scanned a lot more. So we'll be analysing that data later. We've been doing this with very little funding at this point, and we're hoping to improve our funding base because we want to really follow these kids into adolescence and see how they do, because the parents, teachers, everyone's very concerned about them. And of course, identifying novel treatments, and we're collaborating with the Monash Institute of Pharmaceuticals and with the Centre for Neural Engineering here at Melbourne University to look at different types of medications that might impact in different ways to the drugs that we've had to date, because we certainly need better treatments. And this is Stan Skafidis, who's the director of the Central Centre for Neural Engineering. Gersh Channa is involved in neuropathological work, and we've been doing some nice work on glutamate receptors. This is a new toy that they've acquired, a helium-ion microscope, which gets you a resolution far higher than an electron microscope. And Stan was showing me the tip of this, where he told me we were viewing single atoms, which seems to me quite extraordinary. So we're able to look at the surface of cells, and the idea is to look at the receptors on the surface of cells to try and interrogate those receptors in some detail. So I've summarised a lot of this as I've been going, so I won't labour the point, but it's important in disorders like schizophrenia, beginning in adolescence and early adulthood, to really place the findings in context, place them in the context of brain maturation, and to use all that we have at our disposal to try and get a handle on what's going on. And not just to focus on risk, but also to focus on resilience, because resilience factors, neurobiological resilience in particular, may be important and something that we need to look at. And finally, you would have seen some of the advertisements that Melbourne University has had recently of collisions, different disciplines, different faculties coming together, colliding to create something new. I love the metaphor because I think it's exactly the way forward in dealing with something as complex as schizophrenia. Bringing together the biological sciences, the basic sciences, etc., is really a very powerful way forward. There's a lot of people to thank in the work that we've been doing. We collaborate nationally, internationally, with many groups, and I think a picture is always better. This is our group. We had NHMRC program grant, that's Pat McGorry there, and Alison Jung, Ian Hickey, Henry Jackson, a younger version of me. Our autism group with Stan, Gersh, and others, Ian Everall, and some of the areas. This is our clinical unit at Sunshine Hospital. purpose-built as well. And we have a new university facility at Sunshine, which is fabulous. And we're linked into all these other areas. Thank you very much.